So welcome back friends, welcome back in another video tutorial for microbiology with Changnik. So in that video tutorial we are going to discuss about iron uptake mechanism of bacteria. Okay, so remember one thing, all the pathogenic microorganisms have to develop something for their survival. Okay, because remember all the pathogenic microorganisms require free iron to survive inside the host. Okay, but our body fortunately developed in such a way that our bacteria could not find any kind of free iron without any effort. Okay, because most of the iron are complexed, or most of the iron are carried by some iron transporter protein, for example lactoferrin, transferrin, hemoglobin, ferritin. Okay, so as a result bacteria have to develop something so that it can have free iron. Okay, so it have developed some robbers for that. Okay, so that it can rob free iron from those iron transporter proteins. So the robbers that iron that bacteria have developed are called siderophore system. Okay, so siderophore are low molecular weight compounds that have the ability to form complex with ferric iron Fe3 plus and supply it to the cell. So I think this. Okay, so siderophore are low molecular weight compound. It can robbed Fe3 plus iron or free iron. Okay, so siderophore are of two types. One is hydroxamate or phenolate catecholate. Hydroxamate, an example of hydroxamate is ferrichrome found in fungi, and example of phenolate catecholate is enterobactin found in E. coli. Okay, so what are the main role of siderophore? Let's see that picture. So suppose this red marked portion are blood circulation. Okay, so here as you can see there is a lactoferrin Fe3 plus complex, and this is our bacteria. Okay, so it needs Fe3 plus ion, mainly iron, that is ferrous Fe2 plus, for its survival. Okay, so what it should do, it will synthesizes siderophore. Okay, these are siderophores. These siderophore have the ability to simply snatch those Fe3 plus ion from the lactoferrin in that case and form a complex with themselves. Okay, so that is your siderophore Fe3 plus complex. Okay, so now what will be the next step of siderophore? Siderophore will simply carry those Fe2 plus ion in the cell surface. Okay, and in the cell surface there will be a parking lot for siderophore, that is the siderophore receptor protein. Okay, so here the siderophore will bind, and in that case there can be two situations going on. So suppose this is the entrance of the cell and it is the cell surface. So here Fe3 plus siderophore complex is formed. There can be two possibilities. One possibility is Fe3 plus ion siderophore simply leave Fe3 plus ion or simply release the Fe3 plus ion so that it can be easily enter directly inside the bacterial cell. Okay, by some mechanism. And another possibility is the entire siderophore Fe3 plus complex will be transported via a ABC transporter. Okay, and inside the bacterial cell the siderophore will be separated from the Fe3 plus, and Fe3 plus will be converted into ferrous that can be readily used up by pathogenic microorganisms. So that is the basic concept behind the siderophore. Okay, siderophore is low molecular weight protein. This low molecular weight protein will store ferric iron from any iron transport protein. Okay, so after that it will form a complex. So now my question is, how siderophore can snatch Fe3 plus ion from lactoferrin. Can lactoferrin cannot prevent this phenomenon, or there are something another thing going on? So remember one thing, that instead of siderophore there are various kind of proteins that are also working that are surface ferric reductases. Okay, so this protein present on the surface of the bacteria, although we are going to discuss about surface ferric reductase sometime later. Let's come here, hemolysin and cytotoxin. So as I have told you that hemoglobin is the protein that can carry iron as a iron transporter, so that hemolysin is secreted by bacteria and it can destroy hemoglobin. Okay, and there are cytotoxin that will destroy another human cells that are usually involved in carrying the iron, and it free up intracellular iron that can be easily picked up by siderophore and transported inside the bacterial surface. Okay, and inside the bacterial surface it will parked itself in the siderophore receptor protein. After that there can be two possibilities: one, ferric Fe3 plus ion directly transferred inside the cell, okay, or the entire Fe3 plus and siderophore complex become transported via a ABC transporter. So here after that there will be conversion of Fe3 plus into Fe2 plus, that is ferrous iron. Okay, and it is facilitated by surface ferric reductase that is present on the surface of the bacteria. Okay, so three proteins that play the entire iron uptake mechanism of the bacteria. Main player is obviously siderophore. Okay, but before that siderophore, hemolysin, if the iron transporter protein is hemoglobin, or cytotoxin if some other cells are carrying the iron, should be activated. Okay, it will destroy those iron transporter protein and freeing up the free iron. Okay, now this free iron will be complexed with the help of siderophore system, and siderophore will carry those free iron in the cell surface, and there will be siderophore receptor protein. This siderophore receptor protein will bind with the siderophore ferric iron complex. Then it will decide whether it will release the ferric iron for direct transport inside the bacteria, or the entire siderophore and ferric iron is transported via a ABC receptor. Okay, both the two possibility can occur, and inside the cell, surface ferric reductase that is present on the surface of the bacteria will convert ferric iron into ferrous iron that can be readily used up by pathogen. That is the entire concept of iron uptake mechanism of bacteria. So let's learn it theoretically. So siderophore are low molecular weight molecules that are able to form complex with ferric iron and supply it to the cell. These are either hydroxamate or phenolate catecholates. So ferrichrome is the hydroxamate produced by fungi, and enterobactin is the catecholate formed by E. coli. Microorganisms secrete siderophore when little iron is available in the medium. Once the iron siderophore complex has reached the cell surface, it binds with a siderophore receptor protein. That is the parking lot for siderophore. Okay, now the iron is either released to enter the cell directly, or the whole iron siderophore complex is transported into the cell by ABC transporter. In E. coli, the siderophore receptor protein is present on the outer membrane of the cell envelope. We know that E. coli is a gram-negative bacteria, so there will be a outer membrane. So in that outer membrane, siderophore receptor will be present, and when the iron reaches the periplasmic plates, it moves through the plasma membrane with the help of another transporter, and after that conversion of ferrous iron occur. So in every cases you can see there will be conversion of ferrous iron. Okay, that means bacteria actually needs Fe2 plus iron, not Fe3 plus iron, but siderophore will provide bacteria the Fe3 plus form. Then bacteria have to convert those Fe3 plus form into Fe2 plus form with the help of surface ferric reductase. Okay, so again let's revise. So three protein that play the important role: first of all the siderophore protein, then hemolysin or cytotoxin depending on the iron transporter protein, and the surface ferric reductase. First of all, hemolysin or cytotoxin. If hemoglobin is the iron transporter, then hemolysin, and another the cell is carrying the iron, then cytotoxin will come and destroy the iron transporter protein, freeing up the iron. Then siderophore will also come immediately, and it will form a complex with those free iron. Okay, now it will carry those free iron in the bacterial surface, and in the bacterial surface there will be iron receptor protein. Okay, so that is siderophore receptor protein, that is the parking lot for siderophore, that yes, I have reached the home. Now let's enter us or only the iron. Okay, so bacteria now will decide either I need just ferric iron, or whether I can successfully uptake ferric iron only, or siderophore have to guide me more to successfully provide me the ferric iron, right? That means it's just like a delivery system, whether you can take the delivery just coming outside your home, or you can say your delivery boy to please deliver it to my room. Okay, so that is the idea. So if delivery boy instructed, if the delivery boy is instructed to come inside the room, then the delivery boy, that is our siderophore, will come with its delivery, that is the ferric iron, inside the home, that means inside the bacteria, with the help of ABC transporter, and then they become separated, and delivery boy gets its payment. Then this ferric iron will be converted into ferrous iron with the help of surface ferric reductase. Okay, and if the ferric iron can be readily, if the bacteria can readily catch the ferric iron, so it will instruct the siderophore to separate in the siderophore receptor protein, and so that it can take the ferric iron directly. Okay, and otherwise it will instruct the siderophore to come via my gate, that is ABC transporter. Okay, so that is the iron uptake mechanism of bacteria. So I hope this video will be helpful to you, and if it is, please hit the like button, share with your friends, and subscribe to my channel to get more videos like that. And thank you for listening to this class.